Welcome back. In this video we will finish the central dogma, and part of that means we're really going to look at proteins in detail, because proteins are what we care about. And as we saw in the last video, a cell's full-time job is synthesizing the correct protein at the correct time in the correct amount. Whether it needs a structural protein or, now would be a good time to pause and remind me the other two major types of protein functions. So structural is a big, common kind of protein. What are the other two? Go ahead and pause and answer. Hopefully you paused and loudly proclaimed to the heavens, many proteins are enzymes. Enzymes are very important. Enzymes allow a cell to control its internal chemistry and do things like get energy from sugar and do things like build new molecules. That's what enzymes do. And then the other major group of proteins you'd see in bacteria are regulators. And we haven't necessarily looked at what they do, but they would modify the way RNA polymerase works, or they would modify the likelihood of making a protein. And that's all you need to know about that for now. So, what is a protein? Well, as I mentioned in the last video, it is a polymer. It is a chain of amino acids. And there are 20 different amino acids in a typical protein. So we could think of those as the 20 letters in the protein alphabet. And a protein might be 300 amino acids long, and it might have a lot of one amino acid and none of some of the others. But essentially, each amino acid has distinct chemistry. Each amino acid is going to interact with other molecules in its own way. And how that protein is made, which amino acids are where in the chain, determines how it's going to fold into a three-dimensional structure. And it determines ultimately how it's going to interact with other molecules. Will it catalyze a chemical reaction, or will it just stick to another protein? What's it going to do? That's all based on which amino acids are where in the protein. So the structure of the protein has to do with how does it fold into a complex three-dimensional shape. And that's, again, mostly based on which amino acids are where in the chain. But also its function and some other things. So, let's look at some amino acids. You don't have to memorize the amino acids, you don't have to draw the amino acids, but you have to believe me when I say there are 20 in a typical protein. So, amino acids are what make up proteins, and I don't know how much chemistry you've had or if you can interpret these diagrams over here, and it doesn't matter because I'm not going to make you draw or decode any of this. But I just want to show you amino acids each have a part they all have in common, and then they have a part that is different. So I said there are 20 different amino acids. Well, what they all have in common is this nitrogen, carbon, carbon, what we call backbone. This nitrogen is attached to two hydrogens, so it's an amine group, it's reactive. And this carbon is attached to two oxygens, so it's a carboxylic acid group. And the way proteins are made is they just bond this carbon to the nitrogen of the next amino acid. And it has its own carbon, which bonds to the next amino acid of the protein. Let me say that again. So again, to make a protein, what we'd have is a carbon and a carbon and a nitrogen. And what the cell would do is bond this carbon to the nitrogen of the next amino acid. And then this carbon would be bonded to the nitrogen of the next amino acid. So I'm not drawing the hydrogens or the oxygens in here because for this they don't matter, but this is the idea. This amino acid would have something here that's unique. And this one would have something here that might be different if it's a different amino acid, and this one here would have something different. And these squiggles I've drawn are the parts of the amino acids that are unique, and they're the parts that give it their chemical properties. So for our purposes, it doesn't matter what they are. Just know each amino acid has its own kind of distinct chemical properties based on this squiggly part. And no one else would draw it as a squiggle. They would draw it as something like this, because amino acids are relatively small and you can draw their whole structure in this way. So tyrosine is a relatively big amino acid. That's it. That's all tyrosine is. If you take biochemistry, you will have to memorize all of them, and it's more fun than you might think, because the more you learn about the amino acids, the better you understand the differences between them, and you can imagine what they do in proteins and start to imagine the effect they have on the world. And that's fun for nerds. Okay, so I've talked about how a protein is a chain of amino acids, and each amino acid has its distinct chemical properties. Well, again, the way to state that is that the sequence of amino acids, so their order, determines how it folds into its final structure and everything else about it. So what this shows is one way of drawing an amino acid sequence, and they've got these 3-letter codes that are the distinct 3-letter codes for the amino acids. So glycine, I think that's aspartic acid. Is it? Yeah, I think that's aspartic acid. Tryptophan, probably glutamine, leucine, histidine. I could be getting that wrong. It doesn't matter. But it's just telling you which amino acid is where in the chain. Other things it's showing is there are really common ways that amino acids will kind of interact with nearby amino acids. So what these things are showing is what happens if you have this amino acid sequence and you just let go of it. What you might find is certain amino acids would flop around and attach here. They'd kind of stick together. And what we see is kind of distinct, very common arrangements where they might line up and make a sheet, where this would come around here and then this would go around there, and you just end up with kind of a sheet arrangement, or they might make a coil, or they might do something different. And then, so if you look at the 3-dimensional structure of any protein, you'll find these little sheets in there and these little coils. Because again, so this sheet might be, 20 or 30 amino acids total, but a protein might have 300 amino acids total. So it's a really long chain of amino acids that is folded in ways humans cannot guess. So I will always draw proteins as squiggles because that's what we have to think of them as. So if you are curious about how an amino acid might affect a protein, here is a slide about it. Notice up here, this thing, curiosity only. This means you can skip this slide completely if you want to, because you won't be tested on this. And I will often abbreviate this just to CUR. And if you see that up here in a black box, it means you can skip that if you want to. But what I'm telling you is that some amino acids have a charge, and a positive charge on one amino acid will stick to a negative charge on another, and that's one way you can make a protein fold. More commonly, hydrophobic and hydrophilic amino acids will change the way the protein interacts with water, and that will make it more or less likely to fold. Okay, so that was proteins, and I really, really, really hope you remember this. Slide 20. Okay, now we're off to DNA and RNA, nucleic acids. So these aren't chains of amino acids, they're chains of nucleotides. So I'm going to draw that in a minute, but first you're going to pause the video and remind us in the central dogma, how would you use the terms DNA, RNA, and protein? How are they related to each other? So just pause the video, go back, remember how they're related to each other. Okay, hopefully that was straightforward for you. here is where I drew these squiggles. These were amino acids. So this was one amino acid, this was a different amino acid, and this was a different one, and they're bonded together in a chain. So if this chain was 300 amino acids long, it would represent a protein. That's amino acids. They're relatively small. Nucleotides are bigger. And so I won't draw individual atoms. What I'm going to draw is little representations. So I'm drawing circles with P's in them. Those refer to phosphate groups that are bonded together. So nucleotides start out with these 3 phosphate groups. And one of them is bonded to what we'll call a sugar. And that sugar is also bonded to what we call a base. So that's a nucleotide. The sugar can be, it can be ribose or deoxyribose. And it might not shock you to know that RNA is made of ribose and DNA is made of deoxyribose. And that's just a difference of one little part of this sugar. But it has a huge, huge impact on biology when you switch to one version or the other. So the base part can be adenine, which we abbreviate A, or guanine, which we abbreviate G. It could be cytosine, which we abbreviate C, and it could be thymine, or T, or uracil, U. So those are the bases. So DNA and RNA really, just like proteins, they have a language to them. You could think of them as letters in an alphabet that make up some kind of word in their own strange way. But proteins have 20 different letters to make up that language. Nucleic acids, like DNA and RNA, only have 4. They have A, C, T, and either T or U. They have thymine if they're DNA, and they have uracil if they are, what's the word I'm looking for? RNA. So T would be in DNA, U would be in RNA. And that's going to be a difference in this base. So these bases can be one of these 5 possibilities. And so where you saw a protein sequence, well, a protein sequence looks like this with glycine, aspartic acid, tryptophan, glutamine. If I'm getting that wrong, let me know. I don't know why I care if I'm getting that wrong or not. Clearly, I wish I had done better in biochemistry 20 years ago. That's an amino acid sequence of a protein. But what about a nucleic acid? What is the nucleotide sequence of a nucleic acid look like? Well, we can abbreviate it because it's going to look like A, C, C, A, G, T, A, C, T. I'm just making that up, but the whole thing is made of those 4, A, C, T, and G. That would be DNA, and that would just be one molecule of DNA. It's just a long chain of nucleotides. But DNA is usually double-stranded, and that's one of the cool things. So the bases have a property of base pairing, which means an A will always stick to a T or a U on the other strand. So I'm going to draw the opposite strand of double-stranded DNA and show you they will line up like that. And then a C, so if I remember correctly, A will make 2 little bonds to a T, and C will make 3 bonds to a G. DNA is also directional, so I can't stop myself, but you don't have to. Write a little 5' and a 3'. That tells us the direction it goes, the direction it's made in. And so if I make the opposite strand of DNA, I have to follow these rules. But if I know this sequence, I know exactly what the opposite strand would be because of base pairing, and that's essential to how DNA works and how we use it. So this is going to be an A, so it's going to be 5' A, G, T, T, A, C, T, G, G, T, 3' N. So that's some double-stranded DNA with the base pairing we expect. RNA is usually single-stranded. So we don't, it is capable of being double-stranded just like DNA, but messenger RNAs are single-stranded always. And that's most of the RNA we're going to think about. So here's an example of RNA. I have to draw the 5', sorry, but it would look like C, C, G, A, U, U, G, G, C, A, U. 3'. And that's it. That would be a short molecule of RNA. A messenger RNA might be something like 1,000 nucleotides long. And the human genome is made of DNA, and it's something like 3 billion nucleotides long. So this short little sequence I can make up is just nothing compared to how long these things really are. Okay, so, whoopsie-doopsie, where are we? DNA and RNA are nucleic acids. They're chains of nucleotides. So when I say nucleotides, you should be able to think A, C, T, G, U. And that A, C, T, G, or U, A, C, T, G, or U, refers to the base. So if we go back, where I drew this, that's the base. And it would be base pairing with another nucleotide over here. And its base would be forming those little bonds. So those little bonds form there. And, oh man, let me just real quick show you what it looks like when nucleotides form a chain. So this is the base, we'll call it B. This is the sugar, we'll call it S. And these are little phosphates. Basically, this sugar will bond with a phosphate from the next one. And this sugar will bond with a phosphate from this next one. So in each one, each nucleotide has a phosphate, a sugar, and a base. Phosphate, sugar, base. Phosphate, sugar, base. These should be bonded. And this base, again, can base pair with another base that would be over here. So if this was DNA, they would have little bonds, little weak hydrogen bonds between those. If this is hard for you to visualize, don't worry about it. You're not going to have to draw this, but I just like to be complete in the videos because I know there are a lot of people who need completeness. They will not be able to believe something if they don't have all the pieces. So if you are one of those people, you're welcome. And if not, let's move on. Yeah, cool. Yes, right, cool. So here is kind of the magic that translates the nucleotide sequence into an amino acid sequence. So if we go back to the central dogma, what do we call it when proteins are synthesized? We call it translation. And that's because that's the process of taking a sequence that looks like this, of RNA, A, C, G, G, U, A, and turning it into an amino acid sequence, like tyrosine, threonine, serine. I do love serine. So this language of nucleotides that, I've just been doing up here versus the language of amino acids. That's what has to happen when proteins are made. And this table shows you what translation is what. So this is, oh cool, right, this is all the codes. I could have just looked here and known the codes. What are you going to do? Aspartic acid, yeah. What you can do here is find every possible 3 nucleotide sequence of RNA. And you could change these U's to T's to make them a 3 nucleotide sequence of DNA. So every possible 3 nucleotide sequence is in this table. And so what we do is we look, if we have, say, AAU, the first base is A, so it's going to be one of these. The second base is A, so it's going to be one of these. And the third base is U, so it's going to be one of these. That's how you use this table. And I would see this is ASN for asparagine. So if the RNA has this sequence, AAU, somewhere in it, it will have an asparagine in the protein. And a ribosome is this giant... Wow. And a ribosome is a giant complex of enzymes, really, a giant complex of enzymes that builds the protein, and it reads the messenger RNA and knows which amino acid to put next in the chain. It's an amazing process, and we will not look at it in any detail unless you come to office hours and ask me about it, because when you learn how a ribosome works, your head will explode and you'll have to put it back together. So this is the central dogma at its most basic form. What you need to be able to do is tell me, why do these processes happen? Why do we store information in DNA? And I actually haven't told you that yet. I'm about to tell you. Why do we store information in DNA? Why do we have a bunch of proteins instead of a bunch of DNAs? So proteins can have all these different chemistries because of all the amino acids. DNA, let's see, where can I put this? DNA is very stable. So a cell can trust that if it makes DNA, the DNA will last the whole cell's lifetime. So if it puts a code for making a protein in DNA, it will still have that the next day if it wants it. RNA is not. It is unstable. And this is really useful to a cell, because if it decides, I want protein number Q or whatever, I want that protein, I want to make that protein, the first thing it has to do is make a messenger RNA, and then that will be translated by a ribosome, and then the cell will have its protein, but the RNA will still be around. So the cell will keep making that protein. But RNA is unstable, so eventually, within a few minutes or hours, that messenger RNA will disappear and the cell will stop making that protein. That means the cell doesn't get stuck making the same protein forever. It knows, okay, for the next hour I'm going to make this protein. After that I can choose a different protein. So this is a diagram you'll often see related to the central dogma. DNA becomes RNA through a process of transcription. RNA becomes protein through a process of translation. This diagram doesn't tell you what DNA, RNA, protein are. You have to know at the same level of detail in the slides. And you'll notice there are no diagrams, there are no pictures of nucleotides in the slides, because I don't want you to think about their structures. But no, DNA is a chain of nucleotides, and RNA is the corresponding chain of nucleotides. protein is a chain of amino acids. And so you need to be able to tell me translation is done by a ribosome, transcription is done by RNA polymerase. That's what you need to be able to tell me about the central dogma. And that's the end of this video. Central dogma is important enough that we make a long video about it. And there you have it.